Hey guys, it's Mrs. Esser, and it's time to talk about cell transport. So, in class, I had you all pretend to be atoms, and we got up and moved around the room. As you saw, not everyone walked in the same exact path and the same exact motion, but instead, we had atoms running into each other, bouncing off of one another. So, that's showing Brownian movement, the random movement of our atoms. And as I turned the heat up in the classroom, you all moved faster, showing atoms move more in a hotter temperature. And then when I turned it down to make the temperature very cold, we moved very, very slowly, showing that atoms move less in colder temperatures. So the movement of atoms is going to overlap with our cell transport, especially today when we're focusing on our passive transport. So one word I want us to think about is this word diffusion, which is the movement of molecules from high to low concentration. So in this picture, we can see the drop of blue dye and see it slowly diffusing through the container of water. If I had a hotter temperature of water, we would see the diffusion occur faster because the molecules are bumping into one another. If we had it in a very cold water, we would see the dye diffuse very slowly. So temperature is one factor that impacts the rate of diffusion. Another is the concentration of the solute or the thing being dissolved. So the higher the concentration of something, the quicker the solution is dissolved. So think about if I add 2 drops of dye versus if I had a full cup of dye. The full cup of dye with that greater concentration gradient is going to cause the entire solution to turn blue much faster. The last factor that impacts the rate of diffusion is surface area. So the smaller the surface area, the slower diffusion. The higher the surface area, the faster diffusion. So the more surface area, the more those atoms can get and hit each other, causing the diffusion to occur much faster. So here's kind of just showing the diffusion of the red dots. We can relate that to the item in class today, showing the molecules hitting each other and spreading out until they are at equilibrium. So the whole goal of cell transport is just to look at the movement across the cell membrane. So this is one of the most important functions of the cell membrane, to control and to regulate the dissolved molecules of liquid from one side to the other. So here I can see my cell membrane. I see my phospholipid bilayer here. I see outside I have a lot of blue dots, so that'd be a high concentration of this molecule. And inside I just have a couple, which is a low concentration. So we're going to be looking at the differences in molecules from one side of the cell membrane to the other. So a term I'm going to be using a lot today is called concentration gradient. So this is the distribution of particles across the space from high to low. So we can see on the left here, I have a high number of blue dots. Over here, I have a low concentration of blue dots. So same thing if we were in the classroom and I had one student go out into the hallway, I'd have a high concentration of students in the classroom and a low concentration of students in the hallway. So that's a concentration gradient, the distribution or the difference between in the classroom and outside of the classroom. So with passive transport, I want you to think about sliding down a slide. So going down doesn't require any energy, and it's the movement of molecules from a high concentration to a low concentration. So you could also relate this to riding a bike downhill. You don't have to pedal. Gravity is just going to pull you down. You're going from the top of the hill, a high concentration, to the bottom of the hill, which would be a low concentration. So our first type of passive transport is called simple diffusion. And this is the movement of particles across cell membrane from areas of high concentration to areas of low concentration, requiring no energy. So any type of passive transport requires no energy whatsoever. Simple diffusion, it's going to go straight through the cell membrane, straight through that phospholipid bilayer. So oxygen, carbon dioxide, those are molecules that would perform simple diffusion across the cell membrane. The second type of passive transport is called facilitated diffusion. So a teacher's job is to facilitate learning. We help you, we aid you in your learning, but we do not do the learning for you. So kind of the same thing in a cell membrane, except it's a protein that is facilitating the movement of molecules from high concentration to low concentration. So here I can see 2 different types of proteins. So I can see my protein channel, and I have these larger molecules going through the transport protein or the transport channel to an area of lower concentration. Here I have the same thing, a protein moving molecules from high concentration to low. So once again, this does not use energy. It is the movement of molecules from an area of high concentration to low concentration with assistance from a transport protein. No energy required whatsoever. And the last form of passive transport is osmosis, which is just the diffusion of water across a semi-permeable membrane. So semi-permeable means some things can pass through, but not others. So we're going to look at our different types of osmosis, which is probably the biggest part of this cell transport unit. So before we talk about the different types of osmosis, I want you to know what this word tonicity means. So tonic means the amount of dissolved substances. So if I'm drinking tonic water, it's going to be water with minerals in it. So the three types we're going to look at today have to do with these three root words. Hyper, think of a high amount of dissolved substances. Hypo will have a low amount of dissolved substances, and iso means equal or staying the same. So our first one, we have hypertonic. So I remember hyper. When I'm hyper, I drink a lot of coffee, but coffee naturally dehydrates me. Or you could think of drinking an energy drink, which has a high amount of sugar. So hypertonic is going to be when I place a cell in a hypertonic solution. The water is going to rush out of the cell to the area of a high solute, high salt or sugar concentration, and it's going to cause the cell to shrink. So whenever I take a cell and place it in a hypertonic solution, water will rush out of the cell and the cell will shrink. Hypotonic is the opposite. So when I place a cell in a hypotonic solution, it's going to have a low amount of dissolved substances, which means the water amount will be a lot higher. So one thing about this is the solute and the solvent always has to add up to 100. So if I have 60% salt, that means I'd have 40% water. So hypotonic, since they have a low amount of dissolved substances, it means it's going to have a high amount of water. So when I place a cell in a hypotonic solution, my cell is going to swell. I think hippo. My cell is going to swell like a hippo in that solution because all of the water is rushing inside of it. Isotonic, iso means equal, so that means the amount of dissolved substances inside and outside of the cell are the same. So maybe I have 40% solute in the solution and 40% solute in the cell. So that means the water is going to go back and forth at an equal rate. So we call this dynamic equilibrium, meaning the water is moving back and forth, but there's no net change. So here's just a picture of this. So I have a 2% sucrose solution. If I add it to distilled water, remember distilled water, pure, perfect water, that cell is going to swell because it's hypotonic. All the water will rush in, causing the cell to swell like a hippo. In the middle here, I have a hypertonic, which means there is a higher amount of solute in the solution than within the cell. We can see 10% is higher than 2%. So when I add that 2% sucrose solution inside, all the water will leave the baggie to go to the solution. And on the very right, I have isotonic, meaning it's going to be equal proportions of each, meaning dynamic equilibrium, the water going in and out at the same time. So here's just an image I found online where you can see it in an animal cell versus a plant cell. So the only difference is a plant cell has that rigid cell wall, which is going to give structure and support. So for example, if I take an animal cell and I put it in a hypotonic solution, if too much water goes into that red blood cell, it could pop. In a plant cell, it's not going to do that. Instead, we say it has a high turgor pressure, and the water is just going to be pushing out on that cell wall. So just some key terms, turgor pressure and plasmolysis. This is when we're talking about plant cells. So turgor pressure is if I put a plant cell in a hypotonic solution, the water will rush in, expand, push against the cell wall, but it will not break. And then plasmolysis is the opposite. Plasma, think plasma membrane. Lysis, we remember from hydrolysis, means to cut or to break. So this occurs when a plant is placed in a hypertonic solution. All the water rushes out, which could cause the cell to shrivel and possibly die. So just looking at a couple tonicity problems. So here I have a solution with 100% water. Since there's 100% water, I know there's 0% solute, so it's sitting in pure, perfect distilled water. Inside, I have the cell, which is 20% water, which means 80% salt. Water is always going to go to the area of highest salt. So in this situation, my water rushes into the cell because the cell or the baggie is sitting in a hypotonic solution. So water rushes into the cell and it's sitting in a hypotonic solution. Here I have 100% water outside again, so my baggie is sitting in pure distilled water. I have 80% water inside the baggie, meaning there's going to be 20% solute. So water always goes to the area of highest solute, so water will rush into the baggie, and we say the baggie is sitting in a hypotonic solution. Here I have 50% water outside, 50% water inside, which means I also have 50% solute outside and 50% solute inside, because remember the solution always has to add up to 100, and the baggie or the cell always has to add up to 100. So this I would call an isotonic solution. We're going to see the movement of molecules equally in and out of the cell, calling it dynamic equilibrium.